often tell my students, think about what biochemists did before there were pipetters and the pipette men. And so that was, invented at University of Wisconsin and by Henry Lardy,
Oh, really?
who was a biochemistry professor at Wisconsin. And before then, people used glass capillary tubes, and that was how you pipetted 10 microliters. Was that you had a glass tube that was graduated with 1 microliter marks, and you filled that up or dispensed it with a little like a little aspirator type device. So things change really quickly, and it's amazing to me now what students think of as commonplace. I still find fascinating, like RNA-seq or doing whole genome sequencing. I think students now see this as routine. To me, this is still awe-inspiring.
Is there any sort of advice you would give yourself if you could go back to your PhD years?
Yes, so I think probably to get out of the lab more, to be honest. So I think that I think the work-life balance question is something that was not really questioned when I was a graduate student. I think it was just work. That was the end of the sentence. But I think that I think that that's a really important thing to keep in mind, and also to focus on what keeps you happy and motivates you in science. So that's actually something I still did when I was a graduate student, and I think that has been successful for me since.
Welcome to our first episode of the Beckman Bites Podcast, organized by the Arnold and Mabel Beckman Foundation. We'll be diving deep into the research and career interests of Beckman Scholars, Arnold Beckman Postdoctoral Fellows, and Beckman Young Investigator awardees. This podcast should be interesting to all varieties of students and researchers. We'll discuss research experiences, different scientific fields, advice for undergraduates and graduate school, and a lot more. This video will be made available on YouTube and the Beckman Foundation website. So if you know someone interested in science or in the Beckman Foundation, please be sure to share the video with them. All right, so let's dive right in. Here I'm chatting with Professor Aaron Hoskins. Professor Hoskins is an associate professor at the University of Wisconsin-Madison, and his research group is interested in studying pre-mRNA splicing and spliceosomes. Thank you for being here, Professor Hoskins.
No problem. Great to be here.
So can you tell us a bit about your research in splicing and spliceosomes?
Yes, so my lab studies RNA splicing, which is the process in which these intervening sequences in RNA transcripts are removed, and then the flanking sequences are joined together. And this is a really common step in eukaryotic RNA processing. It's carried out by a very large machine called the spliceosome, which is made up of about 100 different factors. Most of them are proteins, but there are also 5 RNAs that are important for the reaction. We study splicing using a lot of different methods. So I would say at our heart, we're biochemists, but we do a lot of genetics. We do a lot of single molecule microscopy work, and we do a lot of chemistry and molecular biology as well.
So have you always been interested in this area, or was this something that started in your postdoc?
Yeah, so this was something that started when I was a postdoc. And so when I went to graduate school, I was still in biochemistry, but in a bit of a different area, so more on enzyme mechanism and looking at chemically complex reactions catalyzed by enzymes. But I was always fascinated by these large machines like the ribosome or DNA polymerase or RNA polymerase. And at the time I was a graduate student, one of the first purifications and characterization of the spliceosome had been carried out by Melissa Moore and also by Robin Reed at Harvard Med School. And what these groups found was that these spliceosomes were gigantic. So in humans, there are about 300 splicing factors that were purified and then identified by mass spec. And when I saw this as a graduate student, it really caught my attention, and that's where I began looking to work on that field as a postdoc.
Yeah, that's really exciting. I mean, I've been told by some other scientists that RNA is kind of like the Wild West of biochemistry, and there's so many interactions and so many effects that RNA have that it's beginning to characterize all the effects and the way that RNA affects all of us is very hard to even begin to ask that question.
Yeah, I think that's true. I think for many years, RNA was seen as just kind of the intermediary between DNA and protein, and it was just thought to be it was just the message. I think it's now clear that there's probably just as much post-transcriptional regulation going on for genes as there is at the gene level, at the transcription level. In addition, RNAs can be post-transcriptionally modified and regulated just like proteins can. So I think this is a really exciting time to be working in RNA.
So can you give a little bit of a background on your career with kind of general day-to-day tasks being a professor in a research lab?
Yeah, so it's mostly reading and writing and talking with people, to be honest. So I spend a lot of time reading various things. They could be manuscripts or papers that I'm reviewing or grants, and I spend a lot of time writing my own manuscripts and papers and grants. And then a lot of time talking with people, and these could be collaborators or my graduate students or undergraduates or postdoctoral scientists in my lab and talking with them about their research. So that's pretty much the constant. I also teach undergraduates, and so when I'm teaching and the semester is going on, I spend a lot of time working in the classroom and working with the undergraduates and working on the classroom activities. Then I think that the final thing which people may not realize that professors do is a lot of administrative work, and so there's a lot of behind the scenes work that I do to make the lab happen, and so a lot of this can be very boring, like making sure purchasing orders go through or that people can travel to the conference that they want to travel to. Or it could be, making sure we meet all the biosafety or chemical safety requirements for the lab. And so a lot of that has to be done on a regular basis too.
Wow, yeah, I wouldn't have imagined that a professor would have to kind of handle administrative tasks like that. I figured secretaries or someone else would handle stuff like that.
Yeah, I think that I think very few professors are able to provide, have that level of administrative support. I think the thing to keep in mind is that you have to you have to provide the salary for administrators, and so that's kind of correlated with a certain career stage, which I'm not quite at yet.
Sure. So can you talk about the education and steps that kind of got you to this spot? Where did you do your undergrad and PhD? And
Yep. So I grew up in Indiana, and I went to undergrad at Purdue University as a chemistry major. And I did lots of different things when I was there, but one of the things I did was research in a biochemistry lab, actually two biochemistry labs. And so that kind of convinced me that I wanted to pursue biochemistry as an area of chemistry more so than other things that I worked on. And so I had lots of diverse experiences, but biochemistry really captured my attention. So after that, I went to graduate school at MIT. So I was there in the Department of Chemistry with Joanne Stubbe. And like I said, I had a very different project than what I work on now. So in graduate school, I was doing mechanistic enzymology. And so we were looking at enzymes that catalyze kind of complicated reactions and trying to understand in detail how those enzymes work. So I worked on enzymes involved in de novo purine biosynthesis. And so I worked on a couple of them. And so one was an interesting amidotransferase reaction. And so this was an enzyme that would hydrolyze glutamine to release ammonia, and that ammonia would travel down this little tunnel that was inside the enzyme and then react with a phosphorylated intermediate that the enzyme forms at a secondary active site, and that would then form a product. I also worked with an enzyme that was a carbon dioxide mutase enzyme, and this was a very unique enzyme because it could form carbon-carbon bonds without using an enzyme cofactor. And it kind of was able to use a protein active site and kind of the chemical properties of the substrate to first cleave a carbon bond and then reform it, or actually cleave a carbon nitrogen bond and then reform it at a different position. And so, we were kind of interested in the nitty-gritty of how enzymes catalyze these types of complex reactions.
And for what got you to move towards from enzymes to RNA? What kind of sparked that change?
Yeah, so I would say is still working on enzymes, and so I'm still interested in chemical transformations and how things occur. I think that for me, what became exciting was not necessarily the complexity of the chemistry that was involved, but the complexity of the kind of surrounding machinery. The spliceosome, chemically, the reaction is pretty straightforward, and we know lots of things which do these types of reactions. But what's unique about the spliceosome is kind of the size and the complexity of the machinery that's involved for doing the reaction, as well as all the cellular regulation that goes on in order to regulate that enzyme. And so that is kind of what captivated me as something, I would say I like to think about things which are complex, and I kind of transitioned away from chemical complexity to maybe compositional and regulatory complexity.
So what are some techniques of analysis that go into characterizing such a complex system like a spliceosome?
Right. So we do lots of different things, and so I would say we kind of separate things, to in vitro and in vivo. And so in vitro, we do typical things we would do in any biochemistry laboratory. So we do enzyme assays, and so we measure things like ATP hydrolysis by various components. We look at splicing of RNA transcripts, and we do that usually by radioactively labeling those transcripts or fluorescently labeling them. And we purify proteins, and we can look at the size of the spliceosome or these various complexes using various tools like sedimentation, centrifugation, because the spliceosome is so big, you can't just run it on an SDS page gel. You need to run it in a gradient and then see where it sediments along that gradient. My lab is kind of unique in that we do a lot of single molecule microscopy of the spliceosome, and so we build microscopes, and we figure out how to attach fluorophores to different components of the spliceosome, and then we're able to watch them function, a single spliceosome function as it splices RNA together. And then the final part, which I don't want to leave out, is that because this machinery is so complex, it's really essential in my field that we go back and do experiments in vivo, because there are certain aspects of the splicing process which you cannot recapitulate in vitro. And so we always try to go back into yeast or other organisms and ask, do what we see in vitro, does it translate to the in vivo reaction?
So with that single molecule fluorescence spectroscopy, can you see the fluorescence in real time, or what is the time scale like with that sort of measurement?
Yeah, so we look at the fluorescence in real time, and the time scale kind of depends on what we're looking at. So if we do a complete splicing reaction, typically we're watching things over the course of 30 minutes to 60 minutes. And in that case, due to some constraints based on the fluorophores we use, we have a time resolution of maybe 3 seconds to 5 seconds. So splicing in vitro is pretty slow, and so that's how we're able to get away with such limited time resolution. We have been interested in very specific steps along the splicing pathway. In those cases, we've isolated components, or we've purified certain RNAs or proteins. We've looked at those at much higher time resolution. And so in those cases, we've done 100 milliseconds or less. Or in some cases, we may even need to go faster than that.
And how fast is splicing in vivo? I imagine it must be way faster than 5 seconds.
Yeah, so that's a very contentious issue. So a lot of people are trying to answer that question right now. And so splicing, the majority of splicing in vivo happens co-transcriptionally, and the time scale in yeast would be about 30 seconds or so, maybe a little bit faster. In humans, the numbers are kind of all over the place, and I think the consensus may be that some genes are spliced very fast, so the RNA products of some genes can be processed very, very quickly. In other cases, the splicing is much, much slower, and this could be due to regulation that's happening, for that particular RNA product, inside the cell.
So what are some opportunities and obstacles in this field besides the timescale problems?
So the biggest opportunity in the field has to do with kind of two recent developments. And so one is cryo-electron microscopy. So there are now cryostructures of many different spliceosome complexes from both yeast and humans. And this is a really, really big event for the field because it allows us as biochemists to begin asking questions about structure and function. And so we can do biochemical experiments that really weren't possible before. And so I'm taking advantage of that, and so are a lot of other people in the field. The other big challenge, or the other big opportunity, is in drug development. And so a lot of people have now realized that inhibiting the spliceosome could be therapeutically very useful for treating a number of human diseases, in particular cancers. And so my lab and many other labs are looking at these inhibitors and trying to discover new ones. And again, the cryo-EM structures really help with that, because now you can begin thinking of designing small molecule inhibitors and get at structural information on where those inhibitors bind. I think the biggest obstacle in the field is a quantitative understanding of the kind of underlying physical parameters that govern regulation of splicing. And so these are things like kinetics and thermodynamics that I think a lot of chemists are familiar with and a lot of biophysicists are familiar with. And they're really easy to get in vitro numbers for those parameters. But what we'd like to do are think about the numbers in vivo. And those are what are going to be relevant to regulating gene expression. And that's a really big challenge for the field.
Right. The biochemical standard state is pretty hard to, is it even possible to computationally predict a lot of things in a complex system like a cell?
At this stage, I think we can predict things based on observations, but not necessarily based on a priori. So not necessarily based on fundamental physical constants. So we can look at patterns of behavior or patterns of gene expression that have been observed and recorded based on a lot of different studies, and then use them to predict what may happen in a given cell. That's a little bit different than saying, well, I know that this splicing regulatory protein has a KD of 100 nanomolar for the substrate, and its concentration inside the nucleus is 1 nanomolar, and the binding constants are so and so. Therefore, we would predict this regulatory event to happen. We're a long ways from getting to that later stage, in part because it turns out human gene regulation is extremely complex, and there's a lot of things we need to know.
Sure, it seems like a pretty challenging field, but one that's very, very important.
Yep, absolutely.
So can you talk a little bit about how the Beckman Foundation has influenced you and what the Beckman Young Investigator Award kind of did for your career?
Yes, so the Beckman Foundation and the Beckman Young Investigator Award kind of opens opportunities for you to begin exploring realms of science that I would not have been able to without the award. And so there are things you always kind of would like to do or are curious about, but then, kind of the real world financial constraints come into play. And the Beckman Foundation kind of gives you the opportunity to kind of explore these areas. And that's a really powerful thing for a lab to do, because it allows us to venture out and try new things. And so certainly in my lab, this means looking at new microscopy techniques, looking at new chemical tools. An area of research in the lab that really began with Beckman funding was our work on spliceosome inhibitors, because this came out of kind of nowhere, and with the flexibility of that type of an award, we can really chase down our intuition. The other part is kind of the Beckman family itself, and so my cohort of Beckman Young Investigators that received the award the same year as I did are all really great scientists and really great people, and so they've been great to get to know over the past few years. And we still keep in touch, and we still talk to one another, and that's really been terrific as well.
Yeah, I mean, it's such a great opportunity for a lot of people. I just remember attending the Beckman Symposium a year ago, and I think the level of research and the level of conversations that you have with different members in a community like that is just fantastic. And So I wanted to shift over to advice that you have for students who are just beginning their careers in science, and if there's any advice that you would, or any conversations that you would normally have with undergraduate students that you think are particularly useful at that stage.
Yes. So one thing I would encourage is to make sure you have kind of diverse experiences. And so that can come in lots of different forms. So for example, many people now do undergraduate research, but maybe instead of working in a research lab one summer, you can work in industry or work in government or do journalism or something. So I think it's a really good idea to sample things while you can, to both figure out what you really like and what you want to do, but also to figure out what you don't like and maybe what you don't want to do, because that can be just as important. The other thing I often remind undergraduates, so as they mature in science and as they get more exposure to different things, is that they shouldn't be afraid to try something new. And I think often people, feel that, just because they're a chemistry major, for example, they shouldn't do undergraduate research in a genetics lab. Or if they're a genetics major, they shouldn't do research in an engineering lab. And I think that's definitely not that's not the case. I think you should be open to new experiences. And I think many labs bring in people from all different sorts of backgrounds. And I think that's really important for people to try and sample these different things.
Do you think you sort of got that experience during your undergraduate and your PhD?
Yes. I think in both cases. And so when I was an undergrad, so my first research experience was in organic chemistry, and so I did an internship during a summer at a pharmaceutical company, and I did kind of synthetic organic chemistry. After that, I began doing undergraduate research on campus in a biochemistry lab. and then the lab I was in happened to move to a different university while I was an undergrad, and so I switched labs, at that time, and I began working in inorganic chemistry. And so I kind of went from, my experiences were, looking at peroxide formation during a certain organic synthesis scheme, so very hardcore organic synthetic chemistry, to doing protein biochemistry and structural biology. And then I moved to platinum chemistry. So I looked at platinum coordination compounds, and kind of those three.
Just have to work on lasers.
Yeah. So I think that diversity kind of convinced me that the one thing that really intrigued me was the biochemistry and thinking about how cells work, and so that kind of sent me towards graduate school. When I was in graduate school, my advisor Joanne Stubby gave me a lot of freedom on my project. And so I ended up doing a lot of different things as a graduate student as well. And so I did a lot of work with fluorescence. I did a lot of, I did things like ultracentrifugation, and I had to chemically synthesize things, and I did a lot of enzyme assays, and I, worked with a friend of mine to do DFT calculations when we were trying to figure out a reaction mechanism. And I think that also made me appreciate that, you can really succeed in science by having a very broad background and kind of, taking a problem and doing whatever kind of it takes to solve that problem, and not limiting yourself to one particular niche of science.
Do you think the value from diversifying experiences comes from the different skill sets you might learn, or does it come from just catching on to something that you're really interested in?
I would say both. I think that, I think it gives you an idea of knowing what's possible. I would say where I'm in my career now, it's kind of hard to be an expert in everything, and I don't think anyone can do that. But what is really useful to know is kind of what's going on in different fields and what the different technologies are that are available. And so I may not be an expert in something now, but if I know enough about technology, then maybe I can collaborate with someone who's an expert. And so you have to be able to kind of connect, the problem you're interested in to the available technologies. And sometimes we have that in the lab, sometimes we can make it ourselves, and other times we, work with other people to make that happen.
I'm sure being a professor, you have to be able to speak so many different languages with probably have 100 different collaborations occurring right now.
Yeah, I was going to say mostly psychiatry.
Interesting.
Yeah.
So what was your perspective on science at the PhD stage, and how has that kind of evolved now that you've done a postdoc and you're a professor now?
Yes, so I think I can answer that question in kind of two ways. One is just science in general, how that has changed, because science is very different now, particularly in biochemistry, than when I was a graduate student. And so when I was a graduate student, this was before next generation DNA sequencing. It was before big data became kind of a thing that people did and did routinely. And it was before things like CRISPR and genome editing. And so all of those have really combined to change biochemistry and the way we approach problems. And so, so I think that the progress that have been made in all these fields are now coming together to make it a really exciting time for biochemistry, because you can now do things which were not even possible 20 years ago when I began graduate school. I think actually I'll stop there. I can't remember the other thing I was going to say.
And I mean, the level of resources now is insane, because I remember a different professor telling me that his PhD was coming up with basically adding the PCR. Like while he was doing his PhD, they were thinking about how to even do PCR, and now PCR is one step in one small part of a project with 40 different resources that you can do to synthesize molecules or start to think about a system?
Oh, absolutely. And I think that's true of any successful technology, is that there takes a lot of blood, sweat, and tears to make something happen and to bring it into reality. And then, as time progresses, these things just become commonplace. And, I often tell my students, think about what biochemists did before there were pipetters and the pipette men. And so that was, invented at University of Wisconsin and by Henry Lardy,
Oh, really?
who was a biochemistry professor at Wisconsin. And before then, people used glass capillary tubes, and that was how pipetted 10 microliters. Was that you had a glass tube that was graduated with 1 microliter marks, and you filled that up or dispensed it with a little like a little aspirator type device. So things change really quickly, and it's amazing to me now what students think of as commonplace. I still find fascinating, like RNA-seq or doing whole genome sequencing. I think students now see this as routine. To me, this is still awe-inspiring.
Is there any sort of advice you would give yourself if you could go back to your PhD years?
Yes. So I think probably to get out of the lab more, to be honest. So I think that I think the work-life balance question is something that was not really questioned when I was a graduate student. I think it was just work. That was the end of the sentence. But I think that I think that that's a really important thing to keep in mind, and also to focus on what keeps you happy and motivates you in science. So that's actually something I still did when I was a graduate student, and I think that has been successful for me since.
Yeah, I mean, I've just started as a first year at Northwestern, but I think one of the things that we've been talking about is, not being the student who works like 14 hours a day for 3 months, but then burns out and has to take several weeks off, but, trying to keep a steady pace going and treating it like a marathon and not a sprint.
Yeah, I mean, I think that one thing I think there are two things to keep in mind. So one is that to become successful in science is hard work. So people who are successful scientists work amazingly hard, and I think people would be shocked perhaps to learn how much the average scientist works. The other thing to keep in mind is that the same, people are individuals, and so this something that may work for one person may not work for everyone. And so, some people, just one of my friends in graduate school would have this highly bursty work experience in lab, where she would work just for a few days really intensely and then take a few days off, and it was always this kind of like all or nothing. And she was brilliant, and she got a lot of stuff done, but that was the way she liked to work. I could never do that, and so I'm an early morning person, so I like to get in early. Then I like to leave and go to the gym and eat dinner and stuff, and then go to bed at like 9 p.m. So I think that that works well for me, but that's not for everyone.
Sure. So were there any challenges that you had in particular after obtaining your PhD, thinking about what science to do now, or did you ever think about going into industry instead of pursuing professorship?
Yeah, so I did not think about industry too much. I did think about working at government labs, and so I think so I always found that a little bit more intriguing than kind of the industry connection. And so these are government labs like MIT Lincoln Labs or Los Alamos or these type of places. And I think that ultimately I kind of decided that I like the energy of working at a university. I like the teaching aspect, and so I really like the interactions I have with undergraduates and graduate students, in particular, kind of watching them mature as scientists when you are with them for several years. That's extremely rewarding.
Yeah, and is there sort of any is there some sort of advice that you give your students as far as thinking about life after the PhD and thinking about whether to pursue one career track or another one?
Yes, so I think ultimately they need to decide on what's going to make them happy. I think that's the most important thing. And so there's a lot of I think pressure about, oh, you need to go into work into a certain type of lab or a lab at a certain location, or you need to get a certain set of publications to be successful. And I don't think that's really true. I think what people should most focus on is what's going to be happy, what's going to make them happy, and where do they see themselves as being a fulfilled person. And that may not involve working at this high-powered lab with like 80 people in it. It may involve working somewhere else. And an example of that is where I did my postdoc. And so I postdoced at Brandeis University, which is a very different environment than MIT or Harvard or Caltech or these other places. But it was nonetheless a place I found really exciting and wonderful to work in. Because they were a small community, but they were really excited about science.
So if you are interested in professorship, would you recommend applying to a lot of different schools and trying not to kind of judge them before applying?
Like I said, again, it depends on what makes you happy. And so, when I was applying for faculty jobs, I had family reasons that I needed to be in the Midwest. And so there were lots of places I could apply to, but I did not because, my priority was something else. And that had to have me placed somewhere in the Midwest. So I think it's entirely an individual decision. And ultimately you need to do what works best for you.
I'm really happy it worked out for you and you're still
thank you.
So in general, how do you handle work-life balance? Does it exist for you? Is it?
Yeah, so I think that the like I said, I think that there's no kind of getting around the fact that I think to being successful in science is a lot of hard work. At the same token, I've I've had some good advice from various people, and when I was thinking about this interview, I was thinking that if I were to phrase this up, it'd be like "Let It Go" from the song from the movie Frozen. Just let it go. Because there's so many things that you can spend your time worrying about and getting frustrated with, and ultimately, I don't think it makes you any more of a happy person or does you any good. And so, I think learning how to just relax and not worry about stuff can then make the non-working hours much more enjoyable.
So that sounds like a really good way of kind of viewing how to be happy with what you've got, at least, and then what you. Well, Professor Hoskins, it's been really nice chatting with you. I'm sure a lot of people will find this interview really helpful, whether they're at the undergraduate or graduate level or postdoc. Is there anything you'd like to kind of say to people who might be watching who are interested in science?
Like I said, I would encourage you just to talk with people who are engaged in science in some way. And so those could be professors at your university, science writers, people who do science policy, patent attorneys, and just learn about the different options there are for careers in science, and then begin exploring those yourself, because often you don't know what you like until you've tried, and sampled a bunch of different things. And so just don't be afraid to get out there and explore.
All right. Well, thank you so much for being on the Beckman Bites podcast, Professor Hoskins. I'd like to thank the Beckman Foundation for allowing us to have this sort of setup and reach an audience in this sort of way with this platform. Again, if you are watching this, please be sure to share this with anyone you know who might be interested in the foundation or interested in just learning about science and hearing the conversation on it. So thank you so much, Professor Hoskins.
No problem. Thank you.